seen that you're already writing on the chat, so welcome to this webinar session. Hi Helena, good morning to you. Hi Ben, hi Hakan. Well, we have people from all over the world. Hi Mohammed from Jordan, Nilum for Turkey, Marcello from Italy. Welcome all of you to this new webinar, okay? And thank you for your patience. We have been waiting 2-3 minutes so everyone can join us. And now we are formally going to start with this webinar. As you may know me, my name is Mauro Giaminorio, and I'm sales manager here at PAT Group for the EMEA region. And I have the pleasure to introduce you for today's webinar, which is about masking and protection against degradation through encapsulation by wet granulation. This webinar, it will be very interesting. We recorded here in our new lab in Barcelona, and we would like to share this case of study and also the videos and everything of this work we have done. Before we start with the webinar, let me inform you about the terms. For your information, this webinar is being recorded, and then we will share it with you. The microphone and camera will be shut down to avoid any interruptions. But please, if you have any questions or comments during the presentation, you have a Q&A chat and where you can do it, and we will answer to you. Yes, and all these questions that are not answered during the webinar, we will do it by email. Yes, as always, any comments or whatever you need, you can just go ahead and write to me. I'm going to write you now my email. Okay, Gabriel, if you would like to go ahead with your presentation, I would like to introduce you.
Okay, thank you very much.
Yes. Hi. Thank you, Gabriel.
Can you hear me, right?
Yes, we can hear you very well.
Okay.
Gentlemen, ladies, I would like to introduce now to our today's speaker. He's Dr. Gabriel Porto. He's our Global Director of Ecovida at PAT Group. He's pharmacist, more than only 35 years experience in the pharmaceutical industry. And he's a specialist in pharmaceutical R&D, has a lot of expertise in production process, quality assurance, quality control, and regulatory affairs. I'm pretty sure many of you have already seen him. Yes. And also we have for today's meeting, Angel Medina. Gabriel, if you can go with the next slide. Thank you very much. Here we have Angel. He's our Ecovida R&D manager, and he was the technical leader of today's case of study. Believe me, this will be a great honor and a truly pleasure to listen both of them carefully. So please, Gabriel, Angel, go ahead.
Okay, thank you very much, Mauro. Hello, everybody. I hope you have a nice day today. Thank you for this opportunity, and I hope it can be of use for all of you. Of course, getting nervous, even if spend many time making this kind of events. It's always a pleasure to speak and to talk to a lot of people. So it's the case. We are a lot of people in the room. So more than 100 I can see. Sorry, I cannot see everything. I can see my presentation. So okay, for today we said we start with a question or like a consideration. What would happen if a highly innovative drug fails? Not because of its clinical efficacy, but because it's not degrades and so it's not able to reach the patient target organ, or because the patient simply cannot tolerate because it's taste or smell and it's awful and not pleasant for him or her. In this presentation, we will see how the wet granulation encapsulation in this case can prevent precisely that from happening, and by masking and protecting the drug. Okay, so let's start with it. So we said that the encapsulation or test masking is a technique designed to neutralize unpleasant flavors and odors in food pharmaceutical and nutraceutical ingredients. Okay. Concerning the active protection, it safeguards functional ingredients from degradation and environmental factors like moisture, temperature, oxidation. And there is a key benefit on it. It enhances product acceptability, stability, shelf life, so make it more pleasant. In terms of encapsulation mechanisms, we say that there are two main approaches. One is the taste masking itself, with the incorporation of or addition of sweeteners, flavors, or specific agents that neutralize or attenuate unpleasant taste and odors. Then the coating of can be made with a polymer, so that's a formation of a barrier that prevents the bitter or undesirable active ingredients from coming into direct contact with the tongue, with the taste buds and olfactory receptors. Okay, so by making a barrier. Okay. So there are then alternative or specific techniques like enzymatic formulation that use that use excipients that reduce flavor release within the oral cavity, or a complexation like the use of cyclodextrins or other complexes that bind or include active ingredient inside, so modulating its sensory perception, right? Then lipidization and emulsion is another specific technique used for this kind of applications, where lipid-based matrices can encapsulate the active ingredient, promoting and controlling release and reducing the taste impact on the oral cavity. Okay. Then there are some specialized techniques. Maybe these are less conventional, but somehow more expensive also. In the last 2 years, significant advances have been achieved in the encapsulation of functional ingredients through the application of new strategies and techniques, such as hot melt, extrusion, and microencapsulation, using different techniques, chemical techniques. Some of them are very difficult to scale up. No, sometimes can be tested at lab scale, but difficult to scale up to the industrial scale. Okay. So, what is the encapsulation process? In fact, no? It's a set of techniques aimed to reducing or eliminating the unpleasant taste or odor of a chemical substance, active or maybe also sometimes excipients, without affecting its efficacy. Okay, and in some cases protecting the functional ingredient against degradation. Okay, so to protect and also to mask. Why is important? Improves product acceptability and extends shelf life by enhancing stability of the active ingredient, particularly in oral formulations such as suspensions, chewable tablets, and powders. Okay. So there are some critical considerations, no, for the process to success in a process of encapsulation. So let's consider them. Stability. Okay, the substance whose taste or odor is to be masked, or this is intended to be protected, must not degrade under encapsulation method employed. So, of course, what you have to do to test that the technology or the composition I use to mask and to encapsulate will not impair on the active ingredient. Then the release and viability. The substance must be released and in a controlled manner at the target site or target organ, ensuring its efficacy. So it must not impair on the release and target objective. Okay. Then it must be safe and tolerable. No, the excipients used must be safe and appropriate for the target population, maybe children, adults, patients in general. Okay. And of course, that must be a compliance in regulatory affairs. No, the excipients must comply with the in general recognized as safe (GRAS) used for food and pharmaceutical regulations and approved in the regulation. Okay. Then let's talk about the kind of technological approaches that can be applied to encapsulation. We have organoleptic methods and we have polymeric coating. It's not one or the other, as we will see. It can be combined, okay? So in terms of organoleptic methods, we can speak about sweeteners and flavors, no? That the like sucralose, aspartame, or some flavors like orange, mint, banana, many of them. That helps to hide or cover unpleasant flavors. Then the effervescent agents like sodium bicarbonate, citric acid, what they do is to enhance mouthfeel. Okay. Then we have bitterness blockers themselves, like adenosine monophosphate or lipoproteins or phospholipids that reduce bitter perception by competing at tongue receptors. Their mechanism is totally different. No, they compete with the receptors in tongue. And sodium chloride, for example, that enhances taste masking in specific formulations. So, of course, it's not everything solved with this the use of these organoleptic methods. Often they must be complemented by other techniques like the polymeric coating to have a greater effectiveness, right? Okay. Then concerning coating with polymers, what we have is the process, in fact, is a direct coating that consists of creating an insoluble physical barrier. So what we perform is a barrier in the mouth to protect the particles of the active ingredient, in this case, using polymers, lipids, or sweeteners also inside, either individually or in combination, as we said, single or multi layers. Okay. The most commonly used of these polymers are HPMC, HPC, povidone, some eudrosheet, and carboxymethyl cellulose. Okay. The function is effective in taste masking, and the coating level, the amount of this ingredient used normally runs in between 10 and 25%, depending on the degree of bitterness of the product and the properties of the coating ingredients. Okay. So, well, after making this general introduction of the different techniques used for masking and protection, we thought that what better than a case study to have as an example of it, no? And so our case study today is encapsulation of fish meal to mask flavor and improve product shelf life. Okay. So we found as a very important and great challenge to use this kind of techniques. So this case study applied to the development of a product by using encapsulation process with the purpose of demonstrating practical and visual what we can do with this kind of techniques. Okay. So once a customer approached to us requesting us a solution to their challenge of masking and projecting fish meal, of course, you can imagine the first reaction was wow, and we imagine how it can smell, and you cannot imagine really. It's very strong, and so we said let's start and let's challenge this by using different techniques we commonly use for other challenges. So First, we'll make a definition of fish meal. So for all of you, the different audience have an idea of this. The fish meal is a product that derived from the processing of fresh fish, okay, which is normally cooked and pressed to extract oil and water from it, then dried and ground into a fine and homogeneous powder, okay, and is terrifically smelly, okay. It's really awful. Um, but in fact, which is the significance of fish meal? The advantages have very high. It's very high in proteins, rich in methionine and cysteine, with high digestibility and elevated biological value. Um, Contains vitamins like B12, choline, niacin, pantothenic acid, riboflavin, minerals. Is a source of calcium, copper, iron, phosphorus, and different trace elements. It's low in fiber and really is very simple and efficient its production. Okay. In the process of fish meal production, we have mainly different sequence steps and very specific. First is heating with where coagulates proteins, breaks down fat deposits, and facilitates the release of oil and water that you want to take out to release from it. Pressing or centrifugation that removes the large proportion of these liquids. Then the separation of these liquids from the mass. Evaporation of the stick water to obtain the fat soluble concentrate. It has a very concentrated mass. And then drying of the material. So the solid press cake, you get like cake of the solids with all the properties, in fact, on it. So, and finally, grinding and storage of the final product that is a powder, right? Okay. Okay. Let's talk about now the encapsulation process of the fish meal we did in this opportunity, and that we reproduced. So, which was the customer expectation? In fact, of course, the objective was to reduce or eliminate the perception of undesirable flavors such as bitterness, acidity, or metallic taste in fish meal in order to make it suitable for consumption. And its smell was the biggest challenge, right? The encapsulation techniques must be integrated with formulation technology in a compatible manner, of course, because we need to obtain something that can be manipulated and used, for example, in a hard capsule. And ensuring that the coated particles, such as those obtained by the fluid bed process, that was one of the first approaches we made, can withstand subsequent manufacturing steps such as compression in a tablet or filling in a hard capsule, as I mentioned before. So they express interest in developing a marine derivative protein concentrate free from odor and taste, with the aim of obtaining a more neutral and consumer-acceptable product, okay? With the properties that itself has, no? So we divided the work of the project in two stages. First, the proof of concept and feasibility, and then the development itself, right? So as we use in any challenge of a new development, new product, first proof of concept and feasibility, and then development. So in the proof of concept and feasibility, what we made was a pre-formulation and definition of appropriate technology. What we said, so we planned different trials aimed at developing the formula and defining the appropriate technology for the process. So we combine both. And first thing you do is to trying to determine which is the more appropriate technology, right? Those includes feasibility testing to validate formula or possible formula and process according to customer requirements. And formulas from stage one were adjusted in stage two to optimize cost, the yield, and performance. Of course, we always focus on that what we are doing, in fact, must be profitable, and so cost, yields, performances, productivity are always on the radar, just to optimize the product. Okay. So then, when they arrived to the development stage, our target was to get the definition of formula, technology confirmation, and the cost evaluation, as we said, during stage two. So the formula is defined and adjusted, and the technology proposed in the previous stage is confirmed and challenged. And the cost considerations were also carried out to ensure the economic feasibility of the process. Okay. So we will enter to the proof of concept and feasibility testing stage one. I think it's okay. Mauro, Stephanie, do you want to stop? Is everything okay? Just tell me, because as I'm not seeing anything, just my
Yes,
presentation.
everything is perfect.
Okay, let's go ahead. So
please go ahead. Yes, we have many questions from our assistants that we are going to reply by email. Many of them are asking if we are going to share this presentation. And yes, we will send the presentation and also the webinar, the video, but they will see it in our YouTube channel and also in LinkedIn. We will post everything, yes.
Okay, and please, Mauro said that we are, of course, open then afterwards to have meetings next week, the following, whatever,
Sure.
Okay? Okay, thank you.
We're starting to have technical questions and comments that we need a little bit more time to clarify. So as you said, Gabriel, the best will be to have a meeting. Please, all the assistants, feel free also to write us, and we can explain in detail any comments that you have.
Great.
Thank you, Gabriel.
Great, thank you. So in terms of the first stage, as we said, we make this kind of proof of concept and feasibility testing. What we did was trying to identify the appropriate technology. So starting with the raw materials, we identified the raw material that we start with. It was a marine protein concentrate, which composition was a blend of 70% fish meal and 30% of soy, processed by spray drying, used as the basis for proof of concept testing, and other masking. So that was our starting raw material composition. Okay. As you can see, it has a certain characterization of it with the brown to ochre color. Of course, strong and intense was the smell, very, very, very strong. Believe me, it was terrible, unpleasant. Angel can say that with his people at the lab. They hate me so much. And the appearance of the powder was with a very varied morphology, not regular, in fact, and with the presence of some impurities, of course. We have then there the results of some analysis like moisture, protein contained, ash, and peroxide value. Okay, as a starting raw material. So we made the experimental design by considering some applicable polymers and some flavors to be used. Then in this table you can see a summary of them, some of them. So what we use as technology was in the different trials, and here we are numbering from 1 to 5, but we make different one of them. So by using high shear mixer, fluid bed dryer, or top spray, those were the focuses technology used. Polymers, we use hydroxypropyl cellulose, povidone, carboxymethyl cellulose that were selected from the pool of possible polymers according to our experience and what we study in as a prior art. And then some flavors like vanilla, cherry, lemon, banana, coffee, etc. So also designed and selected considering the raw material we had. Talking about fish meal, we saw that some notes, like some acidic notes, should be present, and in fact, then we demonstrate it was correct. No, so lemon, you will see that was finally present in the selection. So as you can see, we combine different technologies by using these different polymers to arrive to a conclusion of what was the more appropriate technology and polymer and flavor combinations. So in the evaluation of methods and technologies to be applied, what we decided was, as we said, encapsulation with the CMC, so carboxymethyl cellulose. Then we provide optimal properties for the case study with this polymer. It's a water soluble, it's a derivative of natural cellulose, and its main functions are thickening, it's a thickening agent, stabilizer, and a barrier, very good barrier agent. Okay. It's applied and used in pharmaceutical and cosmetic products, so it's also GRAs, so it's a generally recognized as safe material. It's effective in encapsulation and it's efficient in taste masking. Okay, when combined with other components. Some complementary excipients we used in this development were some absorbent like silica with a very high absorption capacity and whitening agent with, in fact, we used the dioxide titanium in this case. Well, in terms of the method employed for the organoleptic coating was the flavors we used really vary depending on the final application of the product. So as said before, for fish meals, maybe it's not the same when you want to mask other thing. And of course, it's very important the target population, it's children, adults. So these kind of things also are interesting to be considered. In this case, what we want to achieve is in a neutral profile, no, as a result. So, on the characteristic odor and taste of fish meal were effectively masked by using lemon and banana in this case. Okay. In terms of the technology that we selected finally for this case study was one of the most widely used techniques. We know this top spray, so that's why we started from this point, coating in a fluid bed dryer. But in certain cases, also high shear mixer is employed, is used. So we use the different alternatives we have at Ecovida, and the combination of high shear mixer and fluid bed dryer was something proper for this case, and we have the exclusive use of fluid bed dryer with top spray for fish meal applications, and you will see that finally we didn't select this as the better solution. Okay, this is just to review what we have available for when we talk about high shear mixer, what we are talking about, right? Okay, so we are talking about this kind of equipment that enables mixing and granulation with very high efficiency in a very short time. We can operate that in different volumes, 1, 3, 5, 10 liters, and adapting to different requirements and batch sizes, okay, for trials. So Then technology chosen for calibration was the cone mill calibrator, also available at our lab. So it is used for reduction and milling of the agglomerate, the granules in this case. So the idea here by using this equipment is to deagglomerate or to reduce particle sizes, okay? So make it more homogeneous. So typically used for this application, can be used for wet granules. Also, if you change the kind of mesh, you use it for dry granules to reduce them to obtain more uniform particles. Okay, so in this case we use a screen for wet. We have a wet a wet granules. So we selected the more appropriate for this. Then after making the calibration, of course, we had to dry after the wet granules were obtained in the high shear mixer and calibrated in the CPS by using our ESICA fluid bed dryer. This equipment is normally used to or is usable for different targets. One is just drying. That was the case for dry granules that we obtain in the high shear mixer. Then also can be used to make top spray granulation. That was one of the techniques we selected, we challenged for this case study. That is, in fact, this granulation, instead of making it in a high shear mixer, we do it directly on the fluid bed dryer. So by applying the liquid, the binder, directly on the fluid bed dryer. And this machine can also be used for bottom spray. That's to be used for microgranules. It's a totally different mechanism and application, but also very significant, used for encapsulation of microgranules by bottom spray coating. Okay. So let's go now to the experimental work. So where you will be able to see what we did at the lab, trying to reproduce in a very short video how it performs. Okay, let me see, Mauro, Steve, if something runs wrong, then we will stop it and
Yes.
run it again. Okay.
Sure. Gabriel, I think it will be important to mention that the original idea of when we started with this webinar was to do it in live. But then we noticed that the time for all this process and everything will be very, very long. So what we did is to make a video during all the process, and we resume it in 2-3 minutes video. But you are most than welcome to visit our lab and do all this work again together. Yes, but do this webinar purpose, we were limited with the time. But I think it represents very, very well all the work done by Angel, you, and all the pharmaceutical colleagues.
You are right. Yes, you're right. Yeah, it was not possible to make on live. It will be very long and interrupted. So we make this video for all of you. And of course, the idea is to invite you also to visit our lab and to make any question you may have, or comments or whatever. That, of course, more than appreciate it. Okay. Let's go. Well, I hope you enjoy it, and it's just a very short video to show how it performs our case study. Okay, so going ahead with the case study. So in terms of formulation and process development, what we can summarize is the schedule was to match the characteristic taste and odors of the fish meal, sorry, while giving a barrier protection to avoid the degradation. It's a protein, so its humidity is not desired, for example. Then the cellulose derivative polymers were employed for the purpose of coating and making a barrier. And we use a combination of flavors, as you saw, one or two, where lemon and banana were used, including these acidic notes, as I mentioned before, that help to mask the kind of active we are talking about. This approach effectively neutralizes and masks the odor and taste of the product, very characteristic of the fish meal. And we use the different methods and components to achieve the desired outcome. And finally, the polymeric granulation in fluid bed dryer using top spray was tested, as we said, but despite being a widely used technique, really the performance was not totally favorable. Yes. So even if in the case of top spray, you have the advantage of using a single step by making granulation, coating, and encapsulation in only one machine and one step, was not totally satisfactory. Okay. So the granulative masking was inefficient. There was some stickiness of the product and very low grade formation of granules. Okay. So it was decided to move forward with the granulation by using high shear mixer to obtain a better granules and more controlled size of granules and strong granules. Okay. That was satisfactory. And finally, we use high shear mixer plus calibration plus drying as a technique. Results were positive with good organoleptic properties, unaffected in its formulation. So everything was favorable, and the selection of kind of materials were finally making a very stable product and with a reasonable cost. So during the trials, we determined the polymeric granulation with addition of flavors. Finally, were the satisfactory masking properties and results on it. And then the high shear mixer granulation and fluid bed dryer techniques were used, and finally the polymeric coating via top spray was not favorable, as we said, but we found that by using high shear mixer and fluid bed dryer were correct, and we got a neutral flavor on the finished product obtained. Okay. Then we did some optimization trials by trying to reduce costs and trying to, for example, the kind of absorbent we used. We used first one more expensive one, then we use a second one that we found also with good results, but lower price. So we make some optimizing on it, considering that this product will be a mass market product. So it should be necessary to have a very cost-effective product, okay, with quality and appearance. So also in terms of acceptance of the product, we use we add a whitening agent because product looks like not so um, uh, good as an aspect itself, not color. So we adjust also with by using this kind of agent. So finally, the result was a granular product with a neutral taste and optimal balance. Okay. So you will see some pictures of the final formulation, but okay, this is the composition we defined for the product by using of course, as you can see, the main component is the fish meal itself. So it was a challenge to use by using not much of the excipients to mask almost 80% in the composition is the fish meal itself. This is something to highlight because we didn't reduce or we didn't dilute a lot the raw material. We have almost 80% of the raw material present in the formulation. Okay. So let's see a flow chart of the manufacturing process. I will explain it to you. So first we have prepare the binder solution, okay, with the purified water, the binder and the flavors selected, lemon, banana, by mixing them in a propeller, okay, with the propeller, with the propeller steering for about 45 minutes. So with that, we got the binder solution. And at the same time, we were working with the high shear mixer, first deaggregating the components, mainly component, remember that was the fish meal itself. So we make the mixing and during 10 minutes with the rest of the components. We perform a granulation in the high shear mixer by adding the binder solution, and we perform the wet granulation here in the high shear mixer, and then we mesh it and we calibrate it through the Comic calibrator by using a 4 millimeters mesh. And then we put that in the fluid bed dryer to finalize our process and to get the encapsulated fish meal particles. Okay. So let's see the results and conclusion we got with this encapsulation. So in our assessment, we saw that the aspect of appearance, particle size distribution, and organoleptic characteristics, including the sensory panel testing, we challenged some. Of course, the first ones were Angel, Juan, Marta, all our team, because nobody wanted to take the risk of testing. We did it, and then after they saw how it performs, everybody said, "Wow, let's challenge this." So we make a sensory panel testing, and we saw very good results. And of course, what we wanted to see, okay, how it was with the protein content, all the analytical and the chemical results as well, no? Where those were assessed to verify that there were no degradation occurring during the process. Okay. So what we obtained was finally a light pale brown to light pale gray, as you say, undefined color. In fact, say for myself, more gray than brown. And a neutral taste and odor were obtained. and the powder with homogeneous particle morphology after calibrating, as you can see in the picture, and the results after the process, okay? In terms of chemical characteristics, excuse me, you see that also before we had a moisture of 6.5 and protein contains 68, ash 13, peroxide value, and you can see that we could we have no effect in the protein contained on the products, okay, after the process. So in conclusion, the development tests yield satisfactory results and were validated by the client. The customers accept the proposal. It, for us, was very satisfactory to see that they were happy with the results. And the following step was to scale up this process. Then, well, a product was obtained satisfactory and were subject to stability studies for more than 12 months. It was okay, and it maintained the initial quality conditions, so it was acceptable. So, to finalize, we are showing you some of our customers that currently believe in on our services and provision of integrated solutions. Some of them. And to end, we want to show you and looking forward to receive you at our pilot plant in Barcelona. We'll be very glad to receive you, of course, for sure. So you can see here some. Okay, everybody, thank you very much. Thank you very much for your time, for assisting this webinar, and I hope to be of useful for all of you. Any challenge you have, any questions or doubts you may have, technically or whatever. Of course, here we are. Okay. Thank you, everybody.
Thank you very much, Gabriel, for the presentation. It was really interesting, and we also would like to thank you all the assistants to this webinar. You can find on the chat a link with some questionnaire that we appreciate your support answering. And also, we would like to inform you that this webinar is a part of 3 webinars that we are going to make. The next one, it will be on November 25th, and we are going to talk about chewable tablets technology for pets. On the following days, we are going to share with you the presentation, the invitations, so you can register to this event. And we can see we have some questions pending that we are going also to answer by email, as we said before. And again, thank you very much for the time. Thank you for joining us. And remember, we are going to share with you all these presentations by mail. Also, we are going to post it in our YouTube channel and also in LinkedIn. And well, yeah. Thank you. Thank you, all of you. For us, it was really interesting to show the new lab that we have here in Barcelona of some of the services we can provide to support your business. I don't know, Gabriel, if you would like to add something else.
We'll answer all the questions, of course. Sorry for that, but of course we got so many, so we'll do it by reading and contacting you to follow up. Okay.
Thank you, Ibrahim, for your comments. Thank you, Marcelo, Helena, Enrico. A pleasure.
Everybody.
Yes. Ibrahim from Saudi Arabia. from Jordan. Volkan, also thank you very much. We appreciate your comments. There are many colleagues and customers answering.
See you soon.
See you soon. Bye. Thank you.
Bye. Thank you.
Bye-bye.